Okay. Here you go. 15 seconds. Okay. Okay, it's live stream. Let me. You know what? Oh, I can. Let me copy the link. Okay. Okay. Okay, it's live stream. So here, I'm gonna send you the link. I just sent you the link in via instant messaging. Okay. Okay. You're the host again.
All right, great. So, yeah, let me put it on. Hello, everyone. Okay, so we're just waiting for Danilo to join in. He's going to talk today about how do we see molecular world. And it's going to be a very interesting topic. So I hope you'll have a lot of questions. So yeah, I think we're going to start in about like, as I said, like 3 to 5 minutes. So yes, let's see. Bear with me. All right. Hello. Hi, Danilo, you can unmute yourself. Do you hear me, actually? Okay, perfect. Yes. Great. Okay, it's 10:35, so I think we could start soon. So yeah, thank you, Christian and Yuri, for joining us. And we just also went live stream, so whoever is watching there, thank you. This is our third Inspire Shop, and today we're going to talk about how do we see molecular world, which is actually a very interesting topic because it's a very, it goes to the core of science, and it's where the science really kind of started to took off from the discovery of the DNA and so on. It's a very interesting world, let's put it that way. So I hope, yeah, you will, if you have any questions, just jump in, talk, because this is like, it's supposed to be like a conversation way of Inspire Shop. So yeah, we'll be very happy if you can just jump in with a question or with a comment or anything. So without further ado, Danilo, the floor is yours.
Let's see here. Sure.
Oh, an additional thing that maybe I forgot to say is just that I would like to also first introduce Danilo. He's a PhD student at a Cold Spring Harbor Laboratory and Stony Brook University. And he is basically studying how, let's say, RNA splicing, it's a very fundamental understanding of the cell biology. And he's working in Adrian Craner lab that is working on RNA splicing forever, since they're the pioneer in that. And, you know, Danilo can that maybe says a little bit more about it. But yeah,
Yeah.
thanks for being here.
Yeah, thank you for the introduction. So I see we have two audience members extra, right? Well, at least for now. So I would like to know about these people, what they kind of like background they have, because I would like this to be like a conversation, right? So if they can quickly introduce themselves. Maybe first a jury. Or Christian. If they
Christian
could,
was with us already before, I think, last time.
yeah, but like a
and
two
it's
words
good.
about, I mean, what they know about or what they do quickly, like profession, or
Let's see
they could.
if okay,
Already
well,
know.
okay,
Okay.
yeah, we can, it's
So
fine.
because I'm gonna be asking questions, and I designed this like for people who know very little about biology. So let's see how much people know. No.
Yeah, I think it's good.
So my talk is called "How Can We See the Molecular World?" So I'm gonna start with this slide that is asking the questions: what are we made of, right? So you can see here that this is a typical picture of what we call the different levels of organizations of living organisms, right? So in the furthest end here, we have a person, which is an organism, right? And this we can see with our eyes, right? We can sort them out. This is a person, it's not a tree, it's not a cat, right? And that's the level of organism. So we want to see, or we want to know what are they made of, we can clearly see with our eyes that they have arms, they have legs, they have heads, right? If we go one level further down, we can see the organ system. In order for us to see this with our own eyes, we need to open them up, basically, right? Either if it's a tree or if it's an animal or a person, and then you can find different organ systems or different organs that they perform different functions, right? But then if we want to go one step further down or two, to the level of tissues or cells, which are the things that are making the organs and the organ systems, we might need already some tools. For example, the light microscope that hopefully most of you use in high school or in elementary school, right? That these kind of devices use optics and light in order to make things bigger so that we can actually see how the cells look like, what kind of components they have inside. But then we can go even further down to the level of molecules and macromolecules that are making those cells. And in that case, the light microscope is not good enough for us to see what are these structures made of. And so we have different tools, and about these kind of tools are the ones that I want to introduce and for you first to get to know them, and second, so that you know how important they are in order for us to understand how things work in biology and all the things that we can do with these molecules that are making the living matter. So hopefully you will also remember from high school biology is that cells are the basic unit of life. And what I mean by this is like in the whole diagram that we had before with all the different levels, right? Remember atoms, molecule, macromolecules, cells. Starting at cells is when we are talking about things that are alive, right? So if they are, and we're gonna talk about COVID later on, and we can discuss whether COVID is alive or not. But the cells are like conventionally the starting point of life, right? So in this slide, I'm showing you like a lot of different types of cells, and the idea is that you could, and they're all drawn at scale, right? So this line here is 100 micrometers, which is like a measuring unit, right? And you can see that some cells are very small, like bacteria, that you can see here, there is like a very small dot compared to the other cells here. And then some human cells like these motor neurons can be very long and bigger. And some muscle cells can also become very large. And then they also have different shapes. And of course, they have different functions, right? So if I'm going to talk about cells as being the basic unit of life, I'm going to make an analogy of houses as being the basic units of cities, right? And the houses can also look different. So if you remember this story, not from high school, maybe from elementary or from your childhood, this is a picture inspired by the tale of the Three Little Pigs, right? Where they were building different houses made of different materials, right? So in this case, for these houses, you can, as in the previous picture with the cells, you can see that they look very different. And in this case, they're also made of different stuff, right? So if you remember the story, there were houses made of wood, one house made of brick, and one made of hay. And the different composition of these materials give these houses different properties, right? So for example, the house that is made of brick is much more sturdy and it can withstand the attacks by the big bad wolf, whereas the other two houses are much more fragile, right? So when we are talking about the houses as well, we can have different parts of the houses that are specialized in different functions, right? For example, you have the chimney of the house that is useful for eliminating the smoke, right? From inside the house, we have the doors that is going to allow external people to enter into the house. And you have also computers, hopefully inside, that is going to be useful for communication. But now we go back to the parallelism between houses as a city's smallest unit, and we move to our like living organisms. We can also have cells, right? That is what I was just talking about before, right? But what are cells made of? So I would like to know if anyone in the audience would like to answer this question. What, like, in the same way in which we were talking about the different houses being made out of hay, brick, and wood, what do you think cells are made of? Any participant, if they want to raise their hand, except Polona. Like
Maybe.
Christian. Okay, what are cells made of?
Hello.
Hello.
Yes. I think cells are made of organic molecules.
Yeah, organic molecules. Yeah, that's a right answer, right? For example,
Um,
one that you remember.
nucleic acids,
Perfect,
proteins.
exactly. Nucleic
Something like
acids,
that.
proteins, exactly that. So that's the right answer. You got the right answer. So here I'm gonna put one of the first main components of cells is water, first of all, right? Because the cells are aqueous solutions, right? So everything is diluted in water inside these living organisms. But then we have these four main that we call macromolecules or components of living organisms that are carbohydrates, lipids, nucleic acids, and proteins, right? So nucleic acids, like Christian said, you know, for one example, nucleic acid is DNA that stores all the information of the cell. But then you have proteins, lipids, and carbohydrates that are also very important. And one thing that is fascinating about these cells is that in contrast to the houses, remember the houses from the tale that I just showed you before, the houses look very different and they were made of very different materials, right? One was made of wood, hay, and bricks. But in the case of the cells, they can look very different. And here we have, for example, a neuron, a red blood cell, and a plant cell, but they're made of the same materials. If we actually were to take them apart and try to identify which materials are making these different cells, it's all the same. All the living matter is made of the same kind of like molecules, same type of base material. And this is because of evolution, right? So we're here in this branch of the evolutionary tree as animals, right? But we're very close to plants. We are cousins with fungi and other slime molds and bacteria. And we all come from the same ancestor that was something that looks like a bacteria, like a very long time ago. And because those ancestors of ours were made of the same things, then we have to be made of the same things. And this is why it's important, if you think about these words, proteins, carbohydrates, lipids, it's also related to our diets, right? But why is this? Because our cells are like tiny houses that sometimes they need repair, or sometimes they need to build a new house, or new cells. And so we need to take from our environment the same kind of materials that the houses or the cells are made of in order to repair them or divide them, right? So this is why all the things that we eat are actually coming from mostly from living things, either plants or animals. And these things that were living things are then made of the same kind of components that we need to make up our bodies, right? So when we are actually eating, what we're doing is like we are taking apart these materials that comes from either broccoli or chicken that are different proteins. We just chop them in pieces, or the lipids or the carbohydrates, and then inside our bodies we just build up whatever structures we need, like our cells need. For example, there are different proteins that are important for cells to transport things inside the cells, or there are structures in the proteins. There are proteins in the cell that they have the function of communicating with the exterior world or between cell to cell. There are other proteins that we are building up inside our cells that are useful for eliminating waste. And I don't have a good analogy for the couch, but probably we can come up with one.
I have a question from
Yes.
previous slide. So when you were talking about this pyramid of what we eat and everything, so does that mean that when they talk about, everywhere here, oh, you need to eat meat to get proteins, meat, does that mean that, like, actually that also if I'm a vegetarian, I'm getting this, like, the same amount of proteins as if I was eating meat, or would I get the same proteins? What does that mean?
No. So if we go back one slide, which is here, you can see how the plant cells, as the animal cells, are all made of the same: carbohydrates, lipids, proteins, and nucleic acids. And when you are digesting these molecules that you're ingesting, you're actually chopping them into the smallest pieces in order for you to make up for your own proteins. So when you're eating a plant, there's also proteins in the plant, and there's also lipids, and there's also carbohydrates. That's why there's animals who eat only plants, right? For example, cows, right? And what it can be different is what kind of type of protein that they have, but once you are digesting it, you're destroying them. So it doesn't really matter what was the original protein look like. Right?
So you're destroying them on those basic components that are here, and it doesn't really matter what type of food you're eating.
No, as long as you are fulfilling all the necessary requirements, right? You can just eat one kind of food, for example, just red meat all your life. It's not going to be a good diet because you're going to be lacking of other stuff, right? Or if you're only going to eat broccoli, it's also, so this is why it's important that you eat different kind of things. But okay, so again, so what we're basically doing, so what I'm trying to get to here is that these proteins actually are molecules that are going to be inside the cells, and they're going to be specialized in different functions, right? Some of them are going to allow nutrients into the cells. Some of them are going to digest other nutrients or break down nutrients inside the cells. Other ones are going to be important for communication, in the same way in which the different parts of the house have different functions, right? And then another piece of information that is important for you is that you know how cells look different. And in general, the cells that look different are making different proteins. For example, here we have a neuron on top, and you know that the nervous system is very important for communication. So in these kind of cells, you are more likely to find this kind of protein that is called dopamine receptor. And this cell is important for the neurons to communicate one to another, right? Dopamine is what we call a neurotransmitter. So it's a small molecule that gets secreted from one neuron, and it goes to the receptor that is in the next neuron in order to communicate a message, right? But these kind of cells probably are not going to have hemoglobin protein, which is the protein that other cells use to carry oxygen. And this is because these neurons are specialized in communication. So they're only going to produce, so they're going to take proteins from the whatever you eat, take them apart, and then they're going to make up their own structure that they need to fulfill their function, which in this case is this kind of receptors. But in the case of a red blood cell, these cells are in the blood, and they are useful for transporting oxygen from one tissue to the other. So these cells don't need to have these dopamine receptors because they're not involved in this communication, but they do have these hemoglobin proteins that are important to bind oxygen and to bring it from one side to another. And another thing that I want you to pay attention now is how the shape of these proteins, actually the dopamine receptor that you can see here and the hemoglobin alpha subunit is strikingly very different, right? And we can, even though they're made of the same kind of components, the proteins are made of the same, what we call amino acids, their shape, their structure is very different. And this is what it allows them to perform a different function, right? It's in the same way where you can have something made of wood that is good for to make a table, and something you can use wood to make a table and wood to make a chair, right? But this different in structure is what allows you to have a different function. I think Christian is raising his hand. You have a question?
Yes, I had a question.
Yeah.
And in the previous slide, you told us that, we saw that plants and animal cells are made from these molecules or same components. These are carbohydrates,
Yeah.
Nucleic acids, something like that.
Lipids and
But my
proteins.
question is, why do we have to classify food to these that are rich in proteins, others rich in carbohydrates,
Yeah.
if they're all made in the same molecules, if their cells are made in the same molecule?
Yeah, good, very good question. So I think the way to answer is like, because the cells, like these cells, for example, we have here the neurons, right? And the red blood cell. They look very different. They are made out of the same stuff, but then the proportion of the stuff that is in them might be different. For example, if you want to think about a muscle cell, either from a person or from a cow or from a dog, muscles, they need to have a lot of protein inside because these proteins are very important for contracting the muscle, which is that the animals use to move, right? When the animals move, they're using proteins inside, like these kind of things that are gonna make the muscle bigger or smaller, and this allows you to, at the level of an organism, to move, for example, your arm, right? So a muscle cell is a kind of cell that is made, I'm gonna make up percentages here, but let's say it's made of 90% protein and then 10% of the other components, right? But then another kind of cells that we call fat cells, their function is different. So if we can, maybe we can go back to the first slide, sorry, because here I have the drawing of all the cells. So here we have a fat cell, right? These cells, the function of the cell is just to store fat inside in order, so if one day you're gonna be hungry and you're not gonna be able to eat, you're gonna be able to eat from this kind of like fat that you are saving in your body. This is why when you are eating a lot, you are getting fat all around your stomach, for example, me. And because you are creating in this kind of area of your body, a lot of these cells, and their function is to store energy, right? To store fat for a later day when you are gonna not having food, when you're hungry, and then if you cannot find food, you can eat from these kind of cells. So in this case, if we compare the adipocyte, that the function is to store fat, versus the muscle, that the function is to move, the muscle has a lot of more protein because this moving function requires a lot of protein to contract and expand the muscle, right? Versus the fat, it doesn't need so much protein. The function of the fat cell is to store much fat for a later day when you're hungry. So these kind of proteins are gonna have maybe 90% lipids and 20% protein. So if you were just to eat fat, for example, if you decide from now on, I'm only gonna eat fat cells, you're gonna have, all the components, but the proportion of the component is different because these fat cells are very rich in lipids and in fat, right? Versus if you only eat muscle, you're gonna have a lot of protein, right? But you may be lacking any other components. So they are made of the same, but the proportion in each cell might be a little bit different because of the function of the cell, right? Or the same happens to the organisms, right? When you're eating leaves from a plant, it's different. The proportion of proteins and carbohydrates might be a little bit different versus when you're eating seeds, right? In the seeds, for example, when you're eating beans or in the potato, there is a root. There's a lot of carbohydrate because these structures are used for store energy as well, right? So when you're eating a potato, you're gonna find protein and carbohydrate, but carbohydrates in a much higher ratio. But very good question. So going back to where we were, so then going back to the proteins, as I told you before, that the proteins are the ones that are making this kind of functions, for example, door to the cell for the neutrons to come inside the cell, or chimney to destroy waste, or computer to communicate, you can see that the shape of these proteins can be very different, right? And the same way in which when you're making a table or a chair, right, the shape is different. And this is why you sit on the chair and you don't sit on the table, right? Because the chair is made for you to sit for this function. So we have all of these proteins that they are in different cells and they're doing different functions. And what we try to do in my field studies, we're trying to see how they look like, how is the structure in the three-dimensional world. For example, this one, as you can see here, looks very different than this one. So how can we see these kind of structures? Because I told you before that we cannot see with our own eyes, and we cannot see with an microscope either, right? So one way to do this, in order for us to see these proteins, how do they look like, and how do they act, is by a technique that is called X-ray crystallography. So a very simple way for you to explain you this technique is that we try to make crystals out of proteins. So in this part of the figure here, you can see what a crystal is like. So a crystal is going to be made of millions of copies of the same protein. So about more than 1,000 million of proteins, all of the same protein, all put together in the crystal in the same kind of a relative orientation. And then we use one X-rays to shoot at these crystals. And according to the structure of the protein, how this protein is in space, they're gonna make the X-rays change in a different way, generating a different pattern. And with this pattern, we can then build a model of the structure that then we can get the information of how this protein looks like inside this crystal. This might be the hardest slide so far. This is one of the techniques that we use to try to understand how do these proteins look in the real world. So one example of this technique is, for example, the use of X-ray crystallography for the structure of DNA. So here the picture, you have Rosalind Franklin. She was a crystallographer. She was living in the first half of the 20th century. And she took this picture in the same kind of methodology that I just explained in the previous image, right? So she made a crystal of DNA, and then she shot X-rays at this crystal, and then she could see this kind of diffraction pattern, this kind of picture of the crystal. And this is what gave the idea to the scientists that this looks like a double helix, like the DNA is then made of two different strands that are going around each other in a staircase way, right? And why is it important for us to see the molecular world? Is because once we know how the protein or how the DNA looks like, then we can understand how it works, right? We can understand the function of it. For example, when the scientists discovered that the DNA is a double helix, then it was very easy to propose that the double helix could be able to open up in order to generate two daughter double helix. And this is how the DNA gets copied and how the DNA passed from the mother cell to the daughter cell, right? And another example, for example, is the coronavirus, right? Using these kind of techniques by trying to identify the structure of the proteins, in this case in the virus, the scientists are able to model, for example, what are the proteins that are making COVID. So this is, for example, the spike protein, the shape of the spike protein that was solved last year. And how do these, for example, in this case, this virus enter to the cells to do an infection, right? Now, this is a model, for example, of the human cell with these proteins that are in the membrane, that is like the doors of these cells. The function of these proteins is to interact with external agents and make them come inside the cells. And you can see here how the spike protein of coronavirus binds to some kind of proteins in these target cells of the virus. And this interaction is where it's going, this specific interaction between the protein from the virus and the protein from your own cells is going to allow the virus to enter the cell and replicate and infect people, right? So this is what is important for us to understand how these proteins look like in order. So if we can see how the protein from the virus is interacting with the protein in the human cell, we can then design molecules to try to break this interaction or inhibit this interaction so that the virus cannot enter inside. So this is how then this protein change conformation, and it's going to fuse the membrane of the virus with the membrane of the host cell, and then the virus is going to fuse into and enter into the cell to infect. So all of these kind of models are possible because we know how the proteins look like, because of these kind of techniques like X-ray crystallography that allow us to understand the 3D structure of the protein, and then we can model how do they actually work. And then, here, there's other ways to see the molecular world. I just talked to you very briefly about crystallography that requires making up these crystals and then shooting X-ray at them and then seeing the pattern of the X-rays. But there's other techniques that are called cryo-electron microscopy that make use of the electron microscope in order to see very small preparation of these proteins. We also can get structures by another technique that is called nuclear magnetic resonance. And recently this year, a new platform that is called AlphaFold is using artificial intelligence in order to predict the structures of the protein without any, only with the information about the sequence of the composition of the protein, they can predict how is it gonna be what shape is it gonna have. So to end this, I just want to show you this introductory video of this new artificial intelligence technique to predict the structure of proteins. And it lasts like maybe 2 minutes, and then we can discuss about it.
Danilo, I think we cannot hear the sound properly.
We cannot hear the sound?
Can you put it louder, maybe?
You don't hear anything, or?
Maybe it's very, very brief. It's very.
I can talk over it. Do you hear me though?
Yeah, I hear you very well, but the sound I don't hear so well. I don't know about the others. Maybe the others hear it better, but I can't hear it. Can you try again?
Let's try again.
I can hear it, but it's very, very low.
So we have the cells that have the proteins inside the body, right? And they make muscles. And the proteins participate in every process. But each protein has a 3D shape that defines what it does, right? There's all of this amount of millions of proteins, but we only know the structure of some of them.
This is how they fold the proteins?
Yeah, this is the folding of proteins.
Mm-hmm.
That now with this new software, they're being able to just predict by the sequence of the protein how the proteins are going to fall into this. And this software is called AlphaFold. And it's based on the previous structures that were known, like by X-ray crystallography or cryo-EM. So they put the sequence and they come up with some structure, predicted structure. And this is how knowing this structure is going to allow us to make progress in different areas. For example, we can design proteins that digest plastics or to capture carbon from the atmosphere or to treat new diseases. And then last message that I want to see is that we need to still help, because I don't know if you could hear the video, but we only know by experimental methods the structure of like a very small fraction of the proteins that are out there. There are billions of proteins that we have no idea how they look like. And now we have these tools like these softwares to kind of predict how they're going to look like, but we still need a lot of work in order to understand how they look like, how they interact with each other, how they interact with other agents. And we can use this information then to kind of like design things. For example, a lot of drugs that are in the clinics. What are they? Once you know the structure of the protein, for example, a protein that is important for cancer, then you can design another molecule that's going to bind that protein and promote, I don't know, the degradation or turn it off, right? And then in that way, treat a lot of different diseases. So that's about it. I'm sorry that you couldn't hear the video. So well, but it's available in YouTube, and we can give you all the links and all this. The cool thing about this new program that is called AlphaFold is like anybody can, it's a website, so it's a database. So anyone can go and just search the name of the protein, and they will give you a predicted three-dimensional structure that it can be, if you just want to know how does any protein looks like, how do we think that it looks like, it's going to be there and you can see it.
Thank you, Danilo. That was great talk and very informative. And actually, it's very interesting how this is actually important to see the structure, like even in the time of coronavirus and SARS-CoV-2, right? When I believe there was a structure already shown how the protein is binding, right? Or
yeah, but that's
least...
the video that we watched, right?
Exactly.
So we were able to do that because of efforts of structural biologists to try to see these kind of proteins by these kind of techniques like X-ray crystallography, KREM, NMR, and now AlphaFold. And this allows you then to try to understand, this is how the virus actually enters. How can we change this, right? How can we design a molecule that will inhibit the virus line, for example? Yeah?
Yeah, exactly. And also I think it is very interesting because a lot of time, like if you just think about like in the COVID times, how fast the scientists, like how all the scientists step together to try to understand this disease from all different angles. To me, that was fascinating. You got scientists that are working on discovering the new treatments, like you said, right, to stop the interaction. Then you have scientists of molecular biology, structural biology to try to understand the connection and how virus actually enters the cells. They took a microscope, the electron microscope images. So it is kind of amazing how much, what a bulk of literature we actually provided in the last year in comparison to when you showed like to the what it was like 60 years ago or 70 years ago when the structure was first discovered. So in 60 years we came so far. It's, if you think about how long we have been on this planet and how short time the science really bloomed, it's extraordinary. It like blows out my mind. So this is really amazing, and I can't wait for the future developments, and hopefully we're gonna get more scientists. So we need more people.
Well, that's the message.
Exactly. So and it's fun. It's really fun, I think. So yeah, do you? I don't know, Christian, Yuri, you have any questions or comments or anything? I'll be happy to answer.
Yes, thank you. And I would really thank Danilo for this presentation. It's really good. As potential scientists, this is really amazing in different domains. But yes, actually I had some questions from the previous presentation. Mm-hmm. Going back to what you said for Christology, I wanted to ask, as you said that you first create some crystals that are in form of the proteins that you want to analyze. I wanted to ask, normally chemically we know that crystals, this is a characteristics of metals, specifically like group 1 metals. So what is the state of these proteins? If we know that crystals are naturally metals, how do you use this Christology? Simply, what is the state of these proteins that you're going to study? Are they metals? So, and if you can take it with my second question, I was also asking myself on this artificial intelligence. You say that we can predict the shapes of these proteins. Mm-hmm. If so, can we have like, can we believe that if we predict these shapes, it would help us to find like long-term answers for some of these diseases that are really complicated, like this coronavirus, for example. Do you find it possible?
Yeah. Okay. So the first question, crystals. So yeah, the thing that the word crystal actually doesn't mean that it's gonna be a metal or anything. Crystal is like a way in which molecules are together organized in the space. For example, salt. You know salt. There's salt that we can put in the table to condiment our food. If you
Yes.
look at the 1 grain of salt, you can see that it's shiny and that it's like that's a crystal. It's a crystal of salt, right? So crystal, what it means is a crystal is a structure that is made of molecules that are all the same. So it could be all salt or all, for example, diamonds, for example, that are made of carbon, all together in a very structured or organized way, like which means that they are all together and all in the same orientation relative to each other, in a kind of a way that is very organized. And at the beginning of the 20th century, they discovered, or maybe even before, something like that, that proteins are also capable, proteins or DNA and other molecules are also capable, if we put a very high amount of protein in certain conditions, they're going to be able to organize themselves like in the same kind of a structure where they are all in a very similar relative position and bound together. And this is what we call a crystal. So these crystals are made of protein, and the way of getting them is just to increase the amount of protein in a very small space. And they're gonna, some proteins, and that was the problem before we had the artificial intelligence, is that not all proteins are gonna be able to generate this kind of crystals in this kind of like nice way organized. So for the proteins that don't like to form this kind of like regular structures, then it's impossible to do extracrystallography. So yeah. And for the second question, yes. So what we've been doing so far up to like last year is to try to use these experimental methods in order to try to understand how these proteins look like. But we have to either create crystals or take proteins from the natural environments in order to study them by electron microscopy in order to come with a model of the structure. But now that we can actually use artificial intelligence to predict the structure, that means that we are not gonna need to make these crystals anymore or try electron microscopy. We can just only by knowing the sequence of the protein, which means it's like the amino acids that are inside and in which order they are, we can have like a prediction of how this can look like in the real world, right? Of course, prediction is not as good as experimental observation as we can do with crystallography or other methods, but the fact that we already have the prediction, we can already, for example, design molecules that will interact with this, and then try to see if this is going to happen in the real world or not. Or we can already propose much more hypotheses with this kind of like predicted model that we can get now with the artificial intelligence. So that is it already provided a lot of advancement where we can move faster in order to make a new hypothesis, even before we have the actual information from the experimental methods. Yeah.
Okay, thank you very much.
Yeah, hopefully I'll answer some of the questions. So yeah, and the good part is that all of this information is available for everyone for free to search in the internet. So next time anybody hears about any protein, you can just go and search and you can find the prediction of how it may look like, if you want to see it, what kind of shape it's going to have.
Great, thank you so much. That is really nice, and it was a very informative talk. So yeah, I think if there are no further questions, we will conclude this workshop, right? Or Inspire Shop with, like Danila said, please join the board of scientists. It's fun. It's a lot of fun, actually, I must say, going from industry into a PhD in academia. It was a fun transition, and I really like it. Then also for the first, I would like to thank Danilo, and thank you so much for preparing this workshop, and thank you, Christian and Yuri, for joining us. And also, I would like to, maybe just pronounce it. Basically, the next week workshop we're going to have on basically, like, it will be personalized medicine, and what does that mean for the future. For now, we will talk about cancer treatments and how the cancer treatment is designed, how we study cancers, and what does it mean precision oncology or personalized medicine in terms of cancer in the future of our healthcare, which also includes artificial intelligence that we're talking today, and many more. So it's going to be very interesting. So yeah, please join us next Saturday at the same time. And yeah, I wish you all a great weekend and a great week, and see you then next weekend.
See you.
Thank
Bye.
you for coming.